I don't have my sound on. There we go. All right, I assume it said we were recording. So, all right, excellent. That's better. Now I apologize. I had my sound off. So let's go ahead and get started. Today is Thursday, May 7th, and today is our last lecture. We are going to finish up our discussion of the autonomic nervous system. And then it's all tests all the time. We have a test next Tuesday, your lab and lecture exam. Again, the lecture exam, the format is the same format as the previous exam, so no surprise on that. Multiple choice, fill in the blank essay questions. The lab format is going to be the same type of questions. The only difference, instead of seeing them one at a time, they will all be presented at once. So you complete all the answers and submit it, and then you'll have the time for that. Again, you have a nice large window to complete both of those exams, and you can complete them in any order that you want. And then a week later, we come back for the final exam. The final exam is going to be 100 multiple choice questions, and you will have 100 minutes to complete it. 1 minute per question is plenty of time to be able to do that. It will only be available during the class time. I've decided I was encouraged to do it just during our finals time, which is a 2-hour window. But with the extreme circumstances that we're dealing with, that seemed a little tiny bit too restrictive for me. So I'm going to limit it to our class time. But I would encourage you to get started on it sooner rather than later to make sure you have time to complete that. Because there will be no extra time given for any reason. So make sure that you complete it during that time. If there's enough interest, and it looks like I've already got a couple people who have mentioned it, that next Thursday we could have, probably during our normal class time, we could have a review so that we could be available. And again, review is not me standing here and telling you what I think is important. Reviews are question answers. You ask me questions and I'll help you answer those questions, or we'll work on it together. But I would be happy to be available to the class as a review on Thursday. And so if there's enough interest in that, then I will add that to our Confer Zoom schedule, so you'll just access it the same way. It's not required, but if you'd like help to preparing for the exam, the final, we can do that as well. One other thing that came up, and hopefully you've noticed it on the main page, our home page, as you enter our Canvas site, I finally got an answer, some clarification about the excused withdrawal. I was correct in that it is the end of the semester when you have to have done it by. However, their definition of the end of the semester and my understanding of what the end of the semester are two different things. I consider the end of the semester when I submit my grades. That is when those things become official. However, technically, I guess the semester actually technically ends on May 19th. It was ironic, not just for you, because you have a final on that day, but my other class has their final on the 20th, the day after the semester officially ends. So what that means is that if you want to have an excused withdrawal, you will have to do that by May 19th. May 19th is the date that must be submitted by. I will, obviously you guys have the exams on the 12th. My other class has exams on the 13th. So I will be working as best I can to get those graded in a timely fashion. And hopefully I would have the grades done. But even if I get the grades done, and even if you take your final at noon, I need to have all the scores before I can figure out if there's going to be a curve for the final, and then how that final is going to affect your other exams and things like that. I don't believe that I will be able to have that completed in a timely fashion for you to be able to submit your excused withdrawal on time. So unfortunately, I can't tell you what your final grade in the class is going to be. However, hopefully, being the sophisticated student that you are, on Tuesday, depending on how you feel you did on your lab and lecture exam, you might be able to get some sense of whether you think you have a shot of getting the grade that you want in this class moving forward. And again, you can try to make as educated a decision of that. Again, I know, because I will get the emails of people, can you just grade mine so that I can see where I'm at? And that doesn't do you any good, because I need to know all the scores to know what the curve is going to be. So I could give you what your raw points are going to be, but if I don't know what the curve is, I don't know what your actual score is going to be. So just grading your test and nobody else's isn't going to help you, because I don't know what the curve is going to be. So I have to have them all graded, unfortunately, so that I can have the curves. So please don't email me asking me to just grade yours so that you can know what your grade is, because I can't do it, because it won't do you any good. All right, it's not that I don't want to, it's just that's not the way math works. All right, so that is that information. Again, it was a great email that I got from our department chair. Some of it was older information from when this originally gave, but it was the information he got from the district office and from our dean, and so I went ahead and posted all of that on there. So if there were any further questions, some of those things are things we already knew and talked about. You get a full refund for out-of-pocket tuition. It doesn't count as one of your 3 attempts and all those kind of things. But I felt the stuff was worthwhile, and as people are trying to have to make those tough decisions right now, I figured I'd post all of that to let you guys have that as well. All right. Questions on any of that? Excellent. My stunned silence, which was I'd love to get so much. Excellent. All right. Well, stunned silence means that I totally understand everything and we are ready to move forward. So let us go ahead and do that. We left off last time and we were playing with our octopus here. We had talked about the left side of this chart where we had discussed, let me move some of these things around so it's easier for me to play. That goes there, that goes there, and that goes there. Excellent. We had talked about the parasympathetic pathways, and the good news about the parasympathetic pathways is that they were pretty simple and straightforward, pretty basic, pretty linear, I guess would probably be the best way to describe them. That because they are discrete, the pathways are much more linear. As we've already indicated in the sympathetic nervous system, right, our parasympathetic gives us local discrete effects. Our sympathetic gives us big global effects. We see that anatomically by the elaborate branching. There are almost an infinite number of ways we could get from one of these pathways to another in this path. So we have large number of synapses, large number of interconnections, so much more big global effects. But the good news is the way we're going to dissect this information is still similar to what we did in the parasympathetic. We need to know where the cell bodies are located. Where are the cell bodies of the preganglionic neurons? Where are the cell bodies of the postganglionic neurons? Then the second thing we need to know is where their axons go. How does this axon, the preganglionic axon, get from its cell body to a ganglion where it's going to synapse? How does that axon from that postganglionic neuron get to its effector? And so it's still the same type of thing. We're still going to be identifying structures, locations where we find these things, and then putting them together to give us driving directions. Like I said, these driving directions are going to be a little bit more complicated because of the broad overlap of them, but there are still some general pathways. And so those are what we'll be identifying, the general pathways to any one of these effectors that we see over here on the left-hand side. And so basically any of these, we will know the routes. All right. Here, like we did last time, we're able to pull out just the sympathetic version of this. And one of the nice things, one of the things that should jump out at you right from the start is those sympathetic preganglionic neurons. Remember our parasympathetic, some were up here in the brainstem, some were down here in the sacral region of the spinal cord. So remember, from an anatomical standpoint, based on its anatomical divisions where it leaves the central nervous system, we call the parasympathetic nervous system, that branch, the craniosacral. Notice here, every single preganglionic neuron in our sympathetic pathway is going to be located in the lateral gray horn of the spinal cord from T1 to L2. So all of them have the exact same starting point, just within this range. All of them are in the lateral gray horn of the spinal cord, all somewhere between T1 and L2. And so not surprisingly, from an anatomical standpoint, our sympathetic nervous system, right, the anatomical subdivision, is to be referred to as the thoracolumbar, because all of the preganglionic neurons, or if you think about it, our sympathetic nervous system starts in the central nervous system, just in the thoracic and lumbar regions of our spinal cord. So that makes that pretty easy. Now, again, as we can see from the pretty picture here, we know that it is going to branch elaborately, and that gives us those big, wide global effects. But there's something else that this illustration shows us as well. And that is that, again, remember, our preganglionic neurons do not synapse on the effectors. Instead, they synapse on ganglia. And if you notice, we actually have two sets of ganglia here. We have this long, almost chain-like appearing structure, which conveniently enough are what we call the chain ganglia. And we have these ganglia to the side, and because they're to the side, they're called the collateral ganglia. Now again, this is a schematic-like illustration. So what they've done is pulled all these structures to the side so that we can see them more clearly. This is a picture that we have looked at earlier when we were identifying things like the white ramus and the gray ramus and the dorsal ramus, dorsal root, ventral ramus, dorsal ramus. Those were the things we talked about when we were talking about the anatomy of the spinal cord and its branches. But this also gives us a sense of these two types of sympathetic ganglia. The first, as I mentioned, are what are called chain ganglia, or because, as you notice, there are, well, let's say it this way, they're paired ganglia, meaning that there are chains of ganglia on the left side and chains of ganglia on the right side as well. These two ganglia are to the side of the spinal cord. And because they're to the side of the spinal cord, they are also sometimes referred to as the paravertebral ganglia, because they run parallel to the spinal cord, parallel to the vertebral column. And in fact, because we know anatomists hate us, they are also sometimes referred to as trunk ganglia. All of those are appropriate terms. I will try to stick with chain ganglia because it tends to be one of the more common terms, but all of them are acceptable. And again, remember, the key is that these are indeed paired. So there's one on the left and one on the right. These chain ganglia are interconnected. So notice there is axons that connect from one to another, going up and down the chain. And we will talk about it. These ganglia are going to innervate basically two main regions. They innervate all surfaces of the skin. And the other thing they innervate is internal organs that are above the diaphragm. That means obviously the heart and the lungs, but our head is above the diaphragm as well. So that also means salivary glands, lacrimal glands, the mucous membrane of your nose, and so on and so forth. So any internal organ above the diaphragm, and basically all surfaces of the skin, back, front, and appendages. Now, the second type of ganglia technically isn't shown on this illustration. However, if you notice, there are these axons that come out of our chain ganglia and come towards the front of the vertebral column. And notice we can see those same axons coming from the other side as well. And that's because down here, a little lower in the picture, but I'm going to cheat and draw it a little bit higher. Here in front of our spinal cord and in front of our spinal column is the second type of sympathetic ganglion. Right? Again, we said these were the ones to the side, so they are often referred to as collateral ganglia. And notice I only drew one. These are unpaired ganglia. They're collateral ganglia because, again, they're to the side of the chain. But what I like as a term is, again, if you think of their relationship to the vertebral column, They are in front of the vertebral column, so they are also sometimes referred to as, also known as, prevertebral ganglia. These unpaired, located in front ganglia, these prevertebral ganglia, right? If we look at the list of what's here, we've gone to all the superficial surfaces, we've gone to all the organs above the diaphragm. So the only thing that is left, the only thing that they can innervate is the organs below the diaphragm. All right, so they innervate the organs below the diaphragm. They are unpaired, whereas these are paired. There's one other big difference. With these chain ganglia, there are many of them, and they vary from person to person. Not everybody has the exact same number of chain ganglia. But when it comes to the collateral ganglia, there are only and always 3. So every single person has 3 a specific unique collateral ganglia, where there are many of the chain ganglia. This gives us a better sense of the you're right. No, there is an I. I misspelled it. Again, I'm not a good typist to begin with, as it is, but especially if I'm trying to talk and not pay attention, I have to look where I'm keying anyway, and so I'm not looking. So yeah, that is not the first and certainly will not be the last of the spelling errors that I have. All right, now, where do we need to be? So now we've seen the orientation, but again, let's go back to this schematic view because I think it's easier to see the things. And then we'll come back to that previous view when we actually have to identify the anatomy and give our driving lessons. As I mentioned, the first type of ganglia are the chain ganglia, or what are known as the paravertebral ganglia. And there are many of them associated with their location and their relationship to the spinal cord. So there are cervical, thoracic, lumbar, sacral, and coccygeal. And there are many of them. It varies from person to person. I think I say this before, but I will say this. Most people have 3 cervical. And of course, as always, what is the most important word in that statement? Most, absolutely. Notice here, as we look at the pretty picture, there is a superior cervical ganglion, a middle cervical ganglion, and an inferior cervical ganglia. So these 3, most people, the top 3 ganglia are cervical ganglia. In a few individuals, this, and let's put a little, I'll highlight them. The middle and inferior will sometimes fuse together into a single structure. So in a few people, there will just be 2 because the middle and inferior fuse together. But as it turns out, no. And that's a great question. It doesn't affect function because as you can see from the illustration, for the most part, both the middle and the inferior go to the same place. They both primarily innervate the heart and the lungs. So the fact that they fuse together doesn't affect their function at all. All right. The number of thoracic, and I swear I have this on here somewhere, so I'm sure it'll go through it again, is somewhere between like 9 and 12. The lumbar is somewhere around 4 to 5. The sacral, I think, is somewhere between 4 or 5, something like that. Oops, I've got those in the wrong place. There is one coccygeal, and we'll see that one in just a minute, where they actually fuse together at the bottom. But the point is that they vary from person to person. Now, as I indicated, oh, and here we see the pretty picture. Here they are paired. I want to emphasize that point, they're paired. So there's some on the left and there's some on the right. Yes, you need to know these, especially the cervical, because the 3 cervical have specific functions. The rest of them pretty much do the same thing. But these first three have a very important function. So not only do you need to be able to identify them, but it is important that you know their functions as well. So know them on an illustration, know them on a model or chart, and know what they do. The other one you need to know is our coccygeal, because something interesting happens with the coccygeal, as we can see down here at the bottom. The two coccygeal ganglia, remember we have these parallel chains that are going down the sides of our spinal cord, they actually fuse together. And when these two coccygeal ganglia fuse together, they form a structure that is known as the ganglion impar. So these fused coccygeal ganglia that turn our two chains into basically a U-shaped nervous tissue structure is known as the ganglion impar. And like I said, these chain ganglia innervate organs of the head, organs of the limb and skin, and the organs above the diaphragm. Let's talk about those three cervical ganglia that we talked about. The superior cervical ganglia is the largest of all of the trunk ganglia. It conveniently is at the top and is the largest, so it usually makes it pretty easy to find on a model or an illustration or a chart or something like that. And as we can see here, for the most part, it innervates the structures of the head. Smooth muscle, like the eyes, the nose, mucous membranes, the lacrimal, right? The skin of the erector pili muscles, blood vessels, sweat glands, salivary glands. Basically, everything in the head is innervated from branches off the superior cervical ganglia. However, notice we did say that these things branch elaborately. And if you look closely at the illustration, you will see, I keep using green, but green is the color of this. So let's emphasize this way. There is a minor input to the heart. So there is just a little bit of a minor input to the heart from that superior cervical ganglia. But for the most part, the superior cervical ganglia innervates the head. Excellent. One of our paths is clearly delineated. If we need to get to anything in the head, we know we have to pass through the superior cervical ganglion. As I mentioned, the middle and inferior may be separate structures. No, it's not the main one that innervates the heart. That is this one here, or really these two here. Remember, as I mentioned, the middle and inferior are separate chain ganglia, but it doesn't matter if these two cervical ganglia fuse together, because as you can see, they mostly do the exact same thing. And that is they innervate the skin, then more importantly, the internal organs in the thoracic cavity. So the organs above the diaphragm. So they go to the heart, they go to the lungs. So if we're going to the heart or lungs, you have choices. You can either come from the inferior or you can come from the middle, cervical chain ganglia. Both would be acceptable for our driving directions. All right. You can get to Stockton by 99. You can get Stockton by, by 80 West or South or whatever it is, East. I don't know, whatever direction that is, right? 5 or 99. You can get there either way, right? They both go the same direction, parallel to each other. And that's what we have here. Our middle and inferior are just two parallel pathways that go the same place, heart and lungs. All right. And as we talked about, sometimes they fuse together. Well, as I said, we have a ton of other ganglia though, right? Like I said, 9 to 12 thoracic, 4 to 5 lumbar, 4 to 5 sacral, and that one coccygeal. So if you look at this is anywhere between what, 23 and 15, 17, 19, something like that, 18 to 23 ganglia for the rest of the chain. And all the rest of these remaining ganglia all basically do the exact same thing. They all innervate the skin. Right? In fact, they do it via those dermatomes. That same L3 spinal nerve that goes to your arm so you can move it. Well, I guess it wouldn't go to your arm, go to your leg so you can move your leg. or the sensory one that comes from there so that you can feel when someone touches your leg. We also have the sympathetic nerves going there to cause the hair to stand up on end, or to hurt when you pull that when you get your leg waxed, or to sweat when you're running, or to dilate your blood vessels, or all the other things that we talked about that the sympathetic does to our skin. So the cervical ganglia, superior cervical goes to the head, middle and inferior goes to the heart and the lungs, and all the rest of the chain are what basically go to the surface of the skin to control the sympathetic functions of our skin. So if we're getting to a skin, we know it's going to be one of these chain ganglia. And that's pretty simple and straightforward too. Yes, you need to know these functions in that the same way that I said I could ask you to give me driving directions in the parasympathetic nervous system. I could ask you, how does the parasympathetic nervous system innervate the heart? And you had to give me that pathway and tell me where the ganglia are located and tell me what neurotransmitters were released and all of that fun stuff. You're going to have to do the exact same thing in the sympathetic nervous system. So if I ask you for the sympathetic pathway of how the sympathetic nervous system would cause your tears to dry up, or how it would affect your heart, or how it would affect the erector pili muscles of your arm, you need to be able to give me the directions of how to get there. And part of those driving directions is going to be, where does the preganglionic neuron synapse on the postganglionic neuron? I know it sounds intimidating, but there's only 4 real pathways. Of course, you will have to know those 4 sympathetic pathways. And anytime that there's going to be 4 of something, you can be fairly certain that one of those is going to be an essay question on the exam. All right. So, and again, here we see that anatomy. We've looked at it a lot of ways. I like this top-down view as well, because again, it shows this relationship of how those chain ganglia are located lateral to the spinal cord, to the side, which again is why they're called the paravertebral ganglion. And again, notice we see here's our spinal nerve where it comes out. There is our gray ramus, and there is our white ramus. Remember, white is lateral, gray is medial. And remember also, collectively, they are known as the rami communicantes. But what this illustration does not show, so I'll conveniently enough draw it in for it, is that there are axons that are going to come out. Oh, this is just the line. I don't want the line. I want the marker, and I want it to be bigger. There are axons that are going to come out of these chain, and they are going to go to a ganglion that is located here. Why am I making a circle when I can just as easily do it that way? A ganglion that is located in front of the vertebrae. So it's the prevertebral collateral ganglion. And remember, as we mentioned, it's unpaired. So notice it's going to receive input from both sides. Right, the left just goes to the left, the right just goes to the right. I actually have that backwards. This is the left, that is the right. For that, but they're both going to go to that one single collateral ganglion that is going to be located in the front. Nope, and more pretty pictures. Again, this is the one we started with. Again, we see our rami communicantes. Conveniently enough, they've even colored them for us. No, white is lateral, gray is medial. White is lateral, gray is medial. Just like the picture shows. One of our illustrations has it incorrect, but this is the correct orientation, and this is the way that you will know it and understand it. And when we give our driving directions, that will make some semblance of sense as to why. And again, here we have those axons that are going to go on their way to our collateral ganglia. And, oh, spoiler, we now know that those are splanchnic nerves, which is something that we've talked about before. All right, let's talk about our 3 prevertebral, or again, collateral ganglia. There are just 3 and only 3, and everyone gets 3. The superior one is the celiac. The middle one is the superior mesenteric. Wait, if this is the one that is the most superior, let's actually make sure then why the heck is it not called superior, but this one is called superior? This one at least is called inferior. They got that one right. It's the most inferior one. Why is this screwed up? Anyone have any idea? Hazard a guess? Still awake? Well, I will tell you. It turns out these called superior and inferior have nothing to do with their relative locations. Similar, you've got the right idea. They are kind of related to something else, and that's really the key. As we've kind of seen and hinted at in other places, typically where blood vessels go, where blood vessels, let me rephrase that, where arteries go, veins tend to follow right alongside, and nerves tend to follow right alongside them as well. So it turns out in the abdominal pelvic cavity, there are several large blood vessels that come off of the descending aorta. 3 of them happen to have large ganglia sitting right next to them. Well, the very first blood vessel that comes off the abdominal aorta is the celiac trunk. And then there's the superior mesenteric artery and the inferior mesenteric artery. And so all 3 of these are named for the blood vessels they sit next to. So they're not named based on their relationship to each other. They're named for the relationship of the blood vessels they sit next to. So these, the superior mesenteric, inferior mesenteric, and celiac are all named based on their location in relation to a blood vessel. Now, if you notice, we have 3 of them here: celiac, superior mesenteric, inferior mesenteric. That's convenient. And notice again, while there's elaborate branching, if we look closely, we can see the general pathways associated with these ganglia. Notice here in black are the postganglionic neurons. And notice that our celiac ganglion, I can sneak this in down here. I'll rewrite it. Ganglion primarily goes to the proximal digestive organs. Right? That means the liver, that means the gallbladder, that means the stomach, that means the spleen, that means the pancreas. Pancreas, right? All of those structures there are all innervated by the celiac ganglion. Notice our superior mesenteric goes to the small intestine and the proximal part of the large intestine. So all of the small intestine and the proximal large intestine. And notice that our inferior mesenteric goes to the rectum, the urinary system, and the reproductive system. So it innervates the urinary system, the reproductive system, and the rectum and anus. And again, notice also, as we talked about, all of these ganglia innervate structures that are below the diaphragm. All right. Can you see it now, Ashley? Perfect. Excellent. All righty. And I think I say this again on the next page, but notice by diagnosing the illustration, we can make sense of that and we can identify those things, but just by looking at it. So let's go through that again. We'll talk about the splenic nerves in just a minute. So innervates abdominal pelvic organs, right? And again, here we see the pretty picture. Celiac ganglion, again, goes to the spleen, the stomach, the liver, the gallbladder, the pancreas, all of those things. Superior mesenteric goes to the small and the large intestine. Why they've drawn it as two circles, I don't have a good answer for you for that, but these collectively are the superior mesenteric ganglion, and our inferior mesenteric ganglion, again, goes to the rectum and anus, the urinary system, and the reproductive system. All right. So now we know which ganglia, and notice in this illustration, they've kind of given us that relationship nicely. Here we have our chain ganglia. They've only shown it on one side, but we know what's on this side is going to be paired on the other. Here we have that collateral ganglion in front of the spinal cord. And now, using an illustration similar to this, we can start giving driving directions, or more specifically, we can describe the innervation pathways. And the good news is there are 4 basic innervation pathways. So, like I said, on the exam, you are likely to get an essay question that says, describe the sympathetic pathway of how, or describe the innervation pathway by which the sympathetic nervous system would innervate X, whatever X is. And in all likelihood, that X is going to be one of the organs we just talked about. Salivary gland, lacrimal gland, heart, stomach, right? Lungs, spleen, anus, right? Any of those things, we can give those as a destination, and you will have to give the driving directions. Your book actually does a really good job of describing these, and has these pictures are actually from your textbook. Your book does talk about all 4 pathways, but it really only emphasizes 3 of them. So it does talk about 3. Notice we're crossing out 3. We are going to specify 4th. And the 4th one isn't actually on the pretty picture here that we're looking at now, but we will get to that. But what we need is a starting point for our driving directions. And for that, we also need to describe some anatomy. Now, I have been able to, I was playing with this a little bit. So again, this is that picture we were looking at before. And if we start here, we can again kind of try to label some of the features that we have talked about. And again, I'm not going to write them on this one. I've got a different one I'll show that on. Wait, what? I missed that question before. Only come off the same side of the U. Yes. So these only, so if we come back, I guess actually this is the picture we want. Yeah, so notice these axons just come off this side of the chain. These come off of that side of the chain. They'll meet in the middle. Yes. So the chain just come off of that same side. They tend to stay ipsilateral. All right. Starting here, let's identify some of the structures. I'm not going to write the names on this one. I'll write it on the next one, and actually, I think the one after that, in fact. Again, we have our spinal cord. We know our starting point is going to be the lateral gray horn. That is where we find our autonomic neurons. And what, again, is the structural classification of those autonomic neurons? Excellent, multipolar. That multipolar autonomic neurons axon comes out the ventral root and then comes to the spinal nerve, at which point it then has choices. All right. Again, some of those choices are the dorsal ramus and the ventral ramus. Some of those choices are the gray ramus and the white ramus. So we have 4 choices that we need. Now, the good news is we know with sympathetic, we need to get to the chain ganglion. We know one other important piece of information about this preganglionic neuron. We know it's multipolar. We know its cell body is located in the lateral gray horn. What do we know about its axon? What do we know about the axons of the preganglionic neurons? Are they the unmyelinated ones or the myelinated ones? Excellent. I've gotten both answers. Which one is it? Myelinated or unmyelinated? Two myelinated. So it's 2-1. Are they right? Yep, there we go. More and more. Excellent. Myelinated. They are myelinated. So if you're a myelinated axon with those many layers of fat on the surface of your axon, if you are going to travel a pathway, which of the two pathways do you think you're more likely to form? Do you think you're more likely to form a white pathway with all that fat on you or a gray pathway with all that fat on you? Now look at that steak you've got in the fridge defrosting for dinner tonight. What color is its fat? Its fat is white, absolutely. So fat is whitish in color. Those lipids, the phospholipids that those Schwann cells have wrapped around it hundreds of times, make it whitish in color. So it is going to travel the white ramus to the chain ganglia. Again, chains can ascend or descend. And then, as we mentioned, you can leave the chain. And this is where this is, there is a minor error on this picture, not an error, an omission on this picture. This chain actually has 6 structures. Oh, I don't like that yellow. That doesn't show up well. Orange show up better? No, it's dark red. Our chain ganglia actually have 6 different structures coming out of them. 1, 2, 3, 4, 5, and this one that we had to cheat and draw in here, right here, like this. Notice also, if we look at the next picture, again, we've removed that pesky spinal cord and some of those things. Again, it still shows the chain, and it shows how the chains are connected to each other. So some of these axons can go up and down the chain. So again, if we were drawing, as we know, we have a multipolar neuron whose cell body is located here in the lateral gray horn. Its axon is going to come out the ventral root to the spinal nerve. And as we talked about it, to get to a chain, because it is myelinated, let's slap some myelin on it so we remember, it is going to form part of the white ramus, that lateral ramus, into the chain. As we talked about, it can sometimes ascend and form a synapse above. It can descend, form a synapse below. Sometimes it'll synapse right away. Or, and again, this is where our picture is lacking, sometimes it will travel out without synapsing as well. Remember, I mentioned there are 6 structures that can come out of this chain ganglia. 1, 2, 3, 4, 5, and 6. I searched the interwebs this morning, again, granted not for a very long period of time, but to do it long enough to be dissatisfied with what was available to me. However, what I did find, which I did like, notice it's not clear. This is a very small image. And so when I blew up the very small image so that we could see it at its appropriate size so that we'd be able to make sense of it, you can't really read the words. Some things can be made out. We can see that there's a lateral horn here. We can see that says dorsal root ganglion, right? I'm assuming, I don't know what the heck that says. I see a V, it should be ventral, maybe ventral efferent fibers. I don't know what it says. It doesn't matter. But what I liked about it, and I'm going to highlight this, is that this is the one picture I found where indeed our chain has 1, 2, 3, 4, 5, 6 things coming out of it. So if we're going to give our driving directions, we have to identify those 6 things. This one, as we already talked about, the lateral ramus is the white and lateral ramus. This one here, And I'm not going to be able to get my drawing in there, so let's use purple. Whoops, wrong direction. Undo. This one here is our gray, and it is the medial ramus. This one here is just the descending chain axons. And if we have descending, oops, if I spelled descending correctly, descending chain, then not surprisingly, we have, I guess I'll have to come all the way from here, ascending chain axons. And that leaves us with those two. Notice one of these, and let's highlight this one again and change the color this time to dark blue. Notice this one is the one that is going to our prevertebral or collateral ganglion. See, you made me all subconscious about my spelling. All right, collateral ganglion here. And this one that goes to the collateral ganglion, I can sneak it in down here, is what we call the splanchnic nerve. Remember, coming off the sacral portion of the spinal cord in the parasympathetic pathway, we had those things called pelvic nerves, which I also told you were sometimes called pelvic splanchnic nerves. And now you know why. The pelvic splanchnic nerves, P for pelvic, P for parasympathetic. Whereas here, going between the chain ganglia and the collateral ganglia are splanchnic nerves, S for sympathetic. So splanchnic by itself is sympathetic. Pelvic splanchnic by itself, well, not by itself, together, is parasympathetic. But like I said, if that's confusing to you, you may use the term pelvis for the parasympathetic ones. But you still need to know these are splanchnic nerves. And yes, that is unfortunately how you spell splanchnic. And that leaves us with this last one right here. This last one right here, as it turns out, in the sympathetic nervous system is simply called a sympathetic nerve. With these structures, so again, let's add a couple more. Let's highlight a couple more. Our lateral gray horn, our ventral root, and that I'll write out because I've written over it. And right here, this sucker right here is our spinal nerve. Those are almost all. There's a couple more tiny tricky things we need to add to this. But for the most part, those are all or most of the structures we are going to need to know for our pathways. I guess there is one more structure while we're here we should talk about, and that is this structure right here. Notice, while it's got everything we need for our chain ganglia, what it is missing, oops, and I put that up there. Let's move this down here. Is that as part of the spinal nerve, we know the spinal nerve branches to form the gray ramus and the white ramus, but remember it also branches to form the dorsal and the ventral rami, oops, rami, plural, because this rami is the plural of ramus, and we'll just put them together as that one there. So when we are doing our driving directions, this is what I want you to visualize. This is the map. Normally I would draw this on the board and we would fill this in, but I think for this it's going to be more intuitive and make more sense if we write it out in a similar fashion to the way we did it when we were doing it for the parasympathetic nervous system. All right. Questions on this? These are the structures involved, and because this is a pretty picture, I'm going to go ahead and take a picture of this to share that, because I want to erase it now so that we can start our driving directions. All right, so again, before I erase it, any questions on this? Because if this doesn't make sense, like I said, it's about to get a whole lot worse. So any questions on the structures that you see in front of you right here before we start using these structures to giving driving directions of how to get to the body we need to get to? Excellent. Stunned silence. Just what I love. Excellent. All right. So here is our starting point then. Let's go ahead and clear this. Now, as I mentioned, there are 4 basic pathways that you are going to be responsible for. And the good news is every single part of them is the exact same directions. I kind of think of it in terms of Siri, right? If I'm driving from my house in Roseville to pretty much anywhere in the world, the first thing I have to do is I have to get to Douglas Boulevard, right? And I know how to get Douglas Boulevard. My drive into Douglas Boulevard, Siri doesn't have to tell me that. It doesn't have to tell me to turn left out of my driveway, right? And turn right on Home Street, right? And all those things to get to Douglas Boulevard. I can get to Douglas Boulevard just fine. Tell me how to get where I'm going from there, right? Siri could save the first 5 steps. I don't need it. And so the good news is the first 5 steps, and I don't actually know if it's 5, but it sounds good that way. The first portion of our driving directions from all of these are going to be the same. So for all 4 pathways, I'm going to show you here on our illustration our driving pathway. And then from there, we will go on to the rest of the path and how they are going to be different. Now, as I mentioned, for our sympathetic nervous system, we are always starting with a cell whose cell body is located in the lateral gray horn. And of course, we know that cell is the preganglionic neuron. And we know some things about this preganglionic neuron. We know it's multipolar. We know its axon is myelinated. We also know it is going to release acetylcholine when it forms its synapse, and it is going to be excitatory. Those are the things we absolutely, positively, definitely know about our preganglionic neuron. And there's one other important thing we know about our neuron, and this helps us with our pathway. We know it always starts in the lateral gray horn of the spinal cord. And we know it terminates. And again, remember, terminates is just a fancy way of saying forms a synapse in a ganglia. We don't know which ganglion, whether it's a chain or whether it's a collateral, but we know it is going to synapse in one of our 2 ganglia. So that's its job. Its job is to get to those ganglia. So when we start our driving directions, and again, here is my multipolar neuron, whose cell body is located in the lateral gray horn. And I'm giving my driving directions. Its axon starts by going out the ventral root. From the ventral root, it goes to the spinal nerve. And from the spinal nerve, we know that it has 2 choices to get to the chain ganglia, the gray ramus and the white ramus. And because it is myelinated, has all that fat on the surface, we know it is going to go down the white ramus. So every single one of your driving directions starts with that preganglionic neuron in the lateral gray horn, out the ventral root, to the spinal nerve, to the chain, to down the white ramus, and then it reaches the chain ganglion. So every single sympathetic pathway question I could possibly ask you for every single organ in the body, it starts with these 1, 2, 3, 4. Hey, what do you know? 5 steps. Questions on that? All right, excellent. Then here's what we're going to do. Clear this. What does the next slide look like? That one. Okay. And again, here, we'll go ahead and quickly draw it on this one as well. Again, we start with the cell body of that preganglionic neuron, which you know is multipolar. We know its axon is myelinated. We know it comes out the ventral root, spinal nerve, white ramus to the chain. And now here at the chain, right, now you've hit Douglas Boulevard. Now, depending on where you're going, you're going to have different choices. And I'll tell you right now, the choices that you have are 4. 1, you can go up the chain and then synapse. 2, you could go down the chain and you could synapse. 3, you could synapse right away in the chain. Or you don't synapse in the chain at all. You go straight on through the chain without synapsing, and instead you synapse in that prevertebral or collateral ganglion. So those are the different steps that we can have once we're in there. 4 different possibilities. So we've looked at the pretty pictures. We'll look at the pretty pictures again, but let's do these driving directions in words now, and then we'll look at the pretty pictures. So, in fact, actually I lied. Let's go back. I'm going to leave it at this picture. It's a teeny bit earlier than I normally like to take a break, but I said things are going to get worse. Things are going to get dense from here. So I think now a quick decompression would be a good thing. Let's go ahead and take a quick break, get something to eat, get something to drink, check the prices of your turnips, whatever it is you need to do. Meet back here in 10 minutes. It is 12:57, so meet back here at, what would that be, 107, and at 107 we will restart from there. Excellent. All right, meet you back here in 10 minutes. All righty. So, as I mentioned, our goal here is going to be driving directions. So, we need to set up a chart kind of similar to what we did last time, because I think that worked pretty well. So, we will start with, again, our first neuron. And of course, that first neuron, I think for this, we're going to have to be a little bit smaller. So what does that look like? That could work. We know our first neuron is the preganglionic neuron. And as we kind of already hinted at, we know a couple things about it. We know that obviously these are all autonomic motors, so it is multipolar. It is myelinated axon. We also know it releases that they all release acetylcholine, which I am going to abbreviate here. And that acetylcholine is always excitatory. Excellent. And I think I'm going to do that to make space. Perfect. Excellent. We also know along this path that we have a second neuron. That second neuron is the postganglionic. And we know stuff about it as well. It is still an autonomic motor neuron, so of course it is multipolar. However, it has an unmyelinated axon. And remember, because we are in the sympathetic nervous system, most, but not all, will release norepinephrine. A few will release acetylcholine. And again, some will be excitatory. And some will be inhibitory, depending on what the effector is. So I need this to be, why did I, there, that works. Perfect. Okay, so that is some starting information. Let's change the color of those just for the heck of it. Excellent. So we need to know our starting point. And since we're starting with the preganglionic, let's go ahead and go with blue for our starting point. And like we said, the good news is for every single pathway, that starting point is going to be the same. We have that preganglionic neuron in the lateral gray horn of the spinal cord from T1 through L2. So the good news is our starting point for every single pathway we are going to be talking about is going to be the same. All right. Now, and again, if we think of starting point, you can also think of where our preganglionic neuron cell body is located. Location of the neuron cell body. That's what we mean by starting point. Next piece of information we need to know is the path the axon takes. And again, the good news is we at least know the beginning of that. And actually, I want this, now that I think about it. I want that up there. There we go. All right. Path the neuron takes, and that we can bring a little bit lower now. And then the next thing we need to know is where it synapses. And again, if we think about it, where the synapse is, if you think about it, is also where the postganglionic neuron cell body is located. And we'll put that there. We need to know what structures are then formed by its axon, so the path the axon takes. And then, obviously, most important of all, we need to know the effector, because that's our goal. Our goal, after all, is to communicate and control an effector with our sympathetic nervous system. All right? This makes some semblance of sense, hopefully, so far, maybe. No one said anything since I've come on, so it's hard to know if anybody's even back or awake or if you can hear me. Excellent, got some yeses. Perfect, perfect, perfect. All right, so I'm not sure the best way to start this, so let's start easy. The best way to start is in terms, well, there's lots of ways you can think of this, but probably the two easiest ways to think about what we're doing, and I'll put little stars here because it amuses me to do so, will be either to think of it in terms of, where's my stamp? Does that work? Perfect. Where the synapse is taking place, or to think of it in terms of what effector we're getting to. And since the essay on the exam is going to involve the effector, let's do it that way. Let's start easy. One effector, and again, oh, let's actually, since we're doing 4 pathways, the first pathway, the first effectors are going to be the organs above the diaphragm. So our goal for the first pathway is going to be to get to one of the organs above the diaphragm. Remember what that can mean. That can mean something in the head. I'm not sure if this is going to give me enough room, but we'll do it this way anyway. No, it's not going to give me enough room. So I got to bring that back. So in the head, or it could also mean the heart and the lungs. Right? So again, in the head would mean a salivary gland, would mean the lacrimal gland for crying, would mean the smooth muscle of the eye, something along those lines. Now, if you think about that, the next question then becomes, where are we going to synapse? What is the path we're going to take? How are we going to synapse? Now, the good news is we know how we begin. The path always starts, oops, it should be blue, with our preganglionic neuron whose cell body is located in the lateral gray horn of the spinal cord from T1 through L2. And as we know, the path that axon is going to take is going to be out the ventral root. That leads to our spinal nerve. That leads to our, wait, why is that green? I don't want green. I want blue. Our spinal nerve, and then we know it takes the white ramus, and that white ramus is going to bring it to our chain ganglion. And if our goal is to get to the head, do we want to synapse in that chain ganglion? Or do we want to pass by the chain ganglion and synapse in a collateral? Which ganglion allows us to innervate the head? True, we remember, and that's a great point. When we get to the chain, once in the chain, and this is an important thing together, remember, once in the chain, it can synapse right away. It can descend, and it can ascend. And remember, this thing branches globally, so it's likely going to do all the above. The point is, where does it want to synapse? Does it want to synapse in a chain ganglion, or does it want to synapse in a collateral ganglion? So which of those two ganglion does it want to synapse in if our goal is to get to the head? Which ganglion did we say innervated the head? Chain or collateral? Again, the head's above the diaphragm, so is it a chain ganglion or is it a collateral ganglion? Chain ganglion, excellent. And that's the magic word I was looking for. Not just any chain ganglion, but the superior cervical chain ganglion. Excellent. So you are absolutely correct. Our goal, why are you not giving me, that's what I want. So we get to the chain ganglion, and then we are going to synapse in the chain ganglion. And you guys are absolutely right. If our goal is to get to the head, not just any chain ganglion, but we want to synapse in the superior cervical chain ganglion. And if we synapse in the superior cervical chain ganglion, then our postganglionics neuron will leave to innervate the head. And it leaves the chain, the path that that postganglionic neuron takes is out that sympathetic nerve. So we go out the lateral gray horn of the spinal cord, ventral root, spinal nerve, white ramus to the chain ganglion, where we can ascend, descend, come straight across, but we're going to synapse in the chain ganglion, and specifically, we're going to synapse in the superior cervical chain ganglion. Then that postganglionic neuron leaves the chain out our sympathetic nerve to go to the head. And if instead we wanted to go to the heart or the lungs, which chain ganglion would we synapse then? Excellent. We would synapse, let's just go, in either the middle or the inferior chain ganglion. And from there, again, it would be a sympathetic nerve. No, it shouldn't be blue. So delete that. Hold on. Delete. Our postganglionic neuron forms the sympathetic nerve. It forms a sympathetic nerve. And it goes to the effector where it releases its norepinephrine and has its effect. So that is pathway 1. We go to an organ above the diaphragm, either the head or the lungs. For that, we're going to synapse in a chain ganglia, either the superior cervical or the middle or inferior cervical. Oh, I forgot to write cervical in there, so let's sneak that in there. And again, I've got the pretty pictures and the pretty words that'll do this again after we look at it this way. So if this is all getting confusing already, then don't worry, we'll look at it again. We're going to beat this dead horse till it makes some semblance of sense. Middle and inferior cervical chain ganglia, which then are postganglionic neuron, form sympathetic nerves to go to the head, to the heart. Oh, they're supposed to be, that's heart. Good gravy. And lungs. There. Just like old days. No! What just happened? Ah, screw it. There. All right. I liquid papered it away. All right, excellent. So that is path 1. However, if you think about it, there is another path where we synapse in a chain ganglion. We can also synapse in any other chain ganglion. And if we synapse in any other chain ganglion, right, the thoracic, the lumbar, the sacral, then which effector are we going to innervate? What is our second pathway? What's the second place we can go? Where do the rest of those chain ganglia innervate? Excellent. Any structure of the skin. That is correct. Now, again, this path starts the same way. We start with a preganglionic neuron whose cell body is located in the lateral gray horn. Oops. Oh, it's white, that's why. Its axon comes out and again forms ventral ramus, pardon me, ventral root. Spinal nerve, white ramus. All right, to the chain. And again, it can go up, it can go down, it can synapse right away. Any of those, it doesn't matter. And because it's broadly branching, it's probably going to do all of those. But what it is going to do is synapse in one of those other chains. And when it synapses in one of those chains, again, it is going to release acetylcholine. It is always going to be excitatory, and it is going to stimulate that postganglionic nerve. And that postganglionic nerve's axon now has an issue. Our postganglionic axon has to get to the skin. Luckily, we know some friends who are going to the skin. The ventral ramus and the dorsal ramus that came off our spinal nerve, they're going to the skin. So if I'm an axon who also needs to get to the skin from the sympathetic nervous system, then I want to go back out of this chain ganglion and get back to that ventral root. I want to get back to that spinal nerve so I can get to the ventral ramus or the dorsal ramus. And if I remember correctly, my axon is unmyelinated. Luckily, there are two paths from the spinal nerve to our chain ganglion. One was the white ramus, which my brother, the preganglionic neuron, used to get here because its axon is myelinated, but my axon is unmyelinated. So might there be a path that I might want to take to get out to the skin? What route can I take? There you go, perfect. So what that postganglionic neuron is going to do is its axon leaves, no, don't want that to be blue. Its axon is going to leave the chain ganglia and it is going to travel out. It forms the gray ramus to either the dorsal or the ventral ramus. And when it goes out that dorsal or ventral ramus, it can then get to the skin. And that is our second pathway. So again, to get to the skin, we start with a preganglionic neuron whose cell body is located in the lateral gray horn. Its axon goes out the ventral root, out the ventral root to the spinal nerve, through the white ramus to synapse in one of my chain ganglia, where it releases acetylcholine and stimulates the postganglionic neuron. That postganglionic neuron, whose cell body is in the chain ganglions, its axon goes out the gray ramus, forms part of either the dorsal or the ventral ramus, and goes to and innervates the skin. Excellent. That, yep, absolutely. So it kind of takes a U-turn. Absolutely. And remember, the white ramus was lateral. So it went past the turn, came in, and then went back out and crosses it again. So it doesn't actually even do a U-turn. It kind of does a 360. And we'll see that when we look at the pretty pictures of it in just a bit. All right. You can use arrows. Again, arrows are a little trickier to draw on the, so the question was, do you have to write it out as an essay question or do it this way? You are, because of the format when we're doing it online, that you're more limited on the types of artistic things that you're able to do in those essay questions. And again, the one thing you don't want me doing is interpreting your answers. So just having an arrow with the word sympathetic nerve next to it forces me to interpret your answer. Does this person understand that that means that the preganglionic axon forms the sympathetic nerve as it leaves the chain ganglia? Or did they just memorize that there was an arrow with the word sympathetic nerve next to it on there? And I can't, let me rephrase that, I'm not willing to interpret people's answers. I don't want to be in that position where I have to guess what someone actually knows and try to interpret what they've written, right? That's fun. Interpretation is fun in an art class, not in a science class. So I much prefer you use words. If you want to have arrows between those, if arrows are easy to do in that essay format, that's fine, but you're going to be graded for what you write. So make sure that it makes sense, right? I'm abbreviating a little bit here for time and space, but make sure on the exam that you're writing these things out. All right, awesome. And we're halfway home. Any questions on that? All right, notice one other thing as well. We're done with the chain ganglia. All of our synapses are in the chain ganglia, are done already. And that brings us to the other ganglia, the collateral ganglia. And remember, with the collateral ganglia, that's going to be our third pathway. And these are to organs below the diaphragm. Now, remember what this can mean. This can mean, and I'm going to abbreviate here, proximal digest. That's the liver, the spleen, the stomach, the gallbladder, the pancreas, all of those types of things. The small intestine and the large intestine. And again, technically, it's just the proximal. So actually, I have room to stick that in. Proximal large intestine, small intestine, and proximal large intestine. And then remember, the third possibility is, we'll just go rectum, urine, and repo. So we have, and let's get them on the same line. Excellent. And now we can cheat and sneak these out a little bit. I think I have enough room so we can make that work. So our third path is to an organ below the diaphragm, but if I've already kind of given it away a little bit, there is going to be some specificity to this. But once again, the next key is, where do these synapses occur? And in this case, the synapses, and I'm going to switch this to, I'm running out of colors here. Let's use, well, we can, since blue for all the other synapses, we'll leave it blue here. Oh, no, that I want green. Stop that. In this case, what we need to do is we need to be able to synapse in one of our collateral ganglia. So we are going to form our synapse in a collateral ganglion. Luckily, we know that there are 3 of them. What's the superior-most collateral ganglion again? There you go. Celiac. Perfect. Then we also have the superior mesenteric, and we have the inferior. Oops. Mesenteric. Those are the 3 possible places where we could synapse. Luckily, 3 locations, luckily 3 different sets of organs. And it isn't coincidental that we set this up this way, because if you synapse in the celiac ganglion, then you are innervating one of the structures of the proximal digestive system. So if I ask you for the path to the stomach, you're going to synapse in the celiac collateral ganglion. If I ask you the pathway to the small intestine, you're going to synapse in the superior mesenteric ganglion. And if I tell you to go to the bladder, you are going to synapse in the inferior mesenteric ganglion. And conveniently enough, from all of these structures, the axons that come out of our postganglionic neuron all are, once again, simply known as sympathetic nerves. So all of these are sympathetic nerves. sympathetic nerves. So then the only trick to this third pathway, once you know which ganglion go with which organs, is how we get to these ganglion. Now, as we have talked about again and again and again, our driving directions all start the same. We start in the ventral ramus. From the ventral ramus, we go to the spinal nerve. From the spinal nerve, we go to the white ramus. And from the white ramus, we go to the chain ganglia. However, here is where this third pathway is different from the first two. In this case, now, our preganglionic axon, our preganglionic neuron, does not synapse. Instead, what happens is the axon continues past the chain ganglion. It passes through it without synapsing, and it leaves by that structure that we called the splanchnic nerve. And that splanchnic nerve is what travels to one of these, oops, you, or you, or you. It is that splanchnic nerve which travels to one of these four ganglion, and then once here, we form our synapse. And of course, that synapse is going to release acetylcholine, and it is always excitatory. So notice for our third pathway, we pass through the chain and do not synapse there, out the splanchnic nerve, to a collateral ganglia where we synapse on a postganglionic neuron. That postganglionic neuron leaves that collateral ganglia, forming a sympathetic nerve, which goes to its appropriate organ and releases norepinephrine and has its effect. Awesome. And just that simply, we have our third pathway. Questions on that? Again, we'll go through all these again with prettier words, with prettier pictures, so that this can make some semblance of sense at the end of this. All right, excellent. Well, I have the biggest question of all. What the hell is left? We've gone to all the organs above the diaphragm. We've gone to all the organs below the diaphragm. We have gone to all surfaces of the skin. What haven't we gone to? What could this possible fourth pathway involve? Well, it turns out there's something special that the sympathetic nervous system does that the parasympathetic doesn't. Right? One of the things we talked about is it branches elaborately so we get big global effects. Every single cell in our body is practically tingling in anticipation when we're in that fight or flight situation because of that bear with an axe. Part of that every single cell in your body tingling from that sensation is from the nerves. That's a close guess. You're half right. But there you go. We want to get something into those blood vessels. And that thing we want to get into the blood vessels to get to affect every single cell of our body is adrenaline. And so that's our fourth pathway. Our fourth pathway, the one that is unique to the sympathetic nervous system, because our parasympathetic doesn't go there, is to the adrenal gland. And in some ways, this is the most unique path of all. The good news is it starts the same. Pathway 4 starts with ventral ramus to, oops, no, blue. To the spinal nerve, to the white ramus, to the chain ganglion. Just like before. And just like the previous one, our preganglionic neuron does not synapse. Oops. In the chain. Instead, it continues out the chain to the splanchnic nerve. Towards our collateral ganglia. In fact, not just any collateral ganglia, but our preganglionic neuron goes to the celiac ganglion, and it turns out it does not synapse there either. Instead, it continues all the way to the adrenal gland. And our adrenal gland, I'll cheat and draw it here for you. As we have talked about many times before in this class, has an outer candy-coated shell and an inner chewy nougat center. What do we typically call that outer area in that type of an organization? When we have an outer region and an inner region, the inner region is the medulla, and the outer region is the cortex. So you're right. The adrenal gland has two regions. It has a cortex, the adrenal cortex, and it has an adrenal medulla. Well, based on the previous question, guess where we're going? To the medulla of the adrenal gland, absolutely. It goes to the medulla, and there it finally synapses. Once it synapses, it stimulates the postganglionic neuron. And that postganglionic neuron is the one that releases adrenaline and noradrenaline, or epinephrine and norepinephrine, into the blood. And with that, we get our big global response, where every single cell of the body has the potential of being affected by this stressful situation. So notice it still is a two-neuron pathway, just the second neuron is actually inside the organ. Kind of more like a parasympathetic pathway if you think about it. And believe it or not, we've actually seen this. I've got a bunch of other pictures to show you, but let me go back to this one first. Notice here, when we were talking about the pathways through the collateral ganglia, where's my annotation to there? Perfect. No, ventral root, ventral ramus, not the same thing. I'll point those out in just a second. Notice, as we talked about, inferior mesenteric ganglion goes to the reproductive organs, urinary system, and the rectum. Superior mesenteric ganglion goes to the small and large intestine. Notice here with our celiac ganglion, we see how it goes to the proximal digestive system. But if you look closely, notice we can be a preganglionic neuron whose cell body is located in the lateral great horn of the spinal cord, come out the ventral root to the spinal nerve, to the white ramus, to a chain ganglion, pass through that chain ganglion without synapsing, form a splanchnic nerve that goes to the celiac ganglion, does not synapse, and continues all the way to the medulla of the adrenal gland, where there's that little black spot inside of there. That is that postganglionic neuron, and that's the one that releases the adrenaline throughout our body. Now, to ask that last question, let's look at this picture. Remember, our spinal nerve branches 4 times. Spinal nerve forms the gray ramus, the white ramus, the ventral ramus, and the dorsal ramus. So the 4 rami come off of the spinal nerve. Remember, it's the roots, the dorsal root and the ventral root, that come together to form the spinal nerve. So in our pathway, we go out the ventral root to the spinal nerve, to the white ramus. And then, as we talked about, if you're going to go to the skin, then you synapse here in the chain, you go out the gray ramus, and then if you go to the ventral ramus, you can go to an arm, or you can go to the front of your body, your belly button, or something like that. So this over here is the ventral ramus. This back here is the ventral root. All right, excellent. We wrote it all out, but like I said, I have it written all out here. And most importantly, I think to really help us understand it, I've got some pretty pictures to help us to visualize it as well. Again, when we went over the directions ourselves, we focused on what the effector was. But remember, we could also think of it just as importantly as where does the synapse occur. So remember, one possibility is where does the synapse occur? Oh, then well, ventral root would have been the one. Hopefully I didn't. Let's see if I did that. Oh, you're right. I did it twice. So you are correct. Thank you for catching that. And I apologize for the confusion. So let's fix that. There, because again, we want to make sure we have it right. And then root and root. And then here I've got ramus. Here, so here ventral ramus. This is the right. Of course, I spelled ventral wrong, but hopefully that's less confusing. Excellent. Thank you both for catching that. I appreciate that, and I apologize for that confusion. Hopefully that makes more sense now. All righty. Thank you, thank you. I appreciate that. Now, okay. So we focused the first time we went through this on what the effector was going to be. But we can just as easily think of it in terms of where the synapse is going to take place. So again, our driving directions all start the same. We have a preganglionic neuron whose cell body is located in the lateral gray horn. It is a multipolar neuron whose axon is myelinated, and it is going to release acetylcholine, which is always excitatory. That myelinated axon leaves out the ventral root to the spinal nerve, and notice we don't have that here, but let's emphasize that, to the spinal nerve. And from the spinal nerve, it travels down the white ramus, which again is the lateral of our communicantes. And here in the chain, it is going to synapse. Remember, it can ascend or descend first, but it is going to synapse in the chain. Now, which chain it synapses in determines where it's going. If it is a thoracic, a lumbar, a sacral, or the coccygeal, any of those, then that postganglionic unmyelinated axon is going to go out the gray ramus to either the dorsal or the ventral ramus. And what effector is it going to then? To the skin. Perfect. And notice here again, we've done it with the pretty words, but like I said, I think sometimes visualizing this really helps. Oh, I thought I had the pictures here. Okay, no, we'll do this one first. And notice I'm going to leave my words where they are, because again, It starts the same way. Preganglionic neuron in the lateral gray horn. Myelinated axon leaves the ventral root to the spinal nerve, forms the right ramus, where it synapses in a chain ganglia, can travel up, can travel down. However, this time, it can either synapse in the superior or the middle and inferior cervical chain ganglion, and again, superior cervical chain. If it synapses in one of those two, or really three, then its axons, or the unmyelinated axons of our postganglionic neuron, are going to come out and form a sympathetic nerve. And if it is from the superior cervical chain, where is it going to go? Superior. If it's the superior cervical chain, where's it going to go? Excellent. That one goes to the head. If it's the middle or inferior, then you are correct. It's going to go to the heart and or to the lungs. All right. And now, I promise, eventually we'll get to it. Here we go. Here are some of the pretty pictures. I like this pretty picture a lot, but I couldn't find it without the labels. That's why I used the other one. And because, again, it's missing one of our 6 pathways coming out of here, although it cheats and shows it to us later. Notice here, we have our lateral gray horn, ventral roots, right? Spinal nerves, white ramus, gray ramus, chain with its ascending and descending chain, and our sympathetic nerve. Now notice, I want to emphasize the right doesn't do one side and the left doesn't do the other side. They've shown us our 2 possible pathways here by synapsing in the chain. On this side here, on the right, we see that again, ventral root, spinal nerve, white ramus, synapse in a chain. From there, it's got the axon, the unmyelinated axon of that postganglionic neuron is going to leave out the sympathetic nerve, going to the head, going to the heart, going to the lungs. Or we can start in the lateral gray horn, out the ventral root to the spinal nerve, down the white ramus, synapse in a chain ganglion. Again, notice this time they actually showed it ascending, but notice both for the first and for the ascending pathway, they show the same thing. We again synapse on that postganglionic neuron. We again release acetylcholine. It is again excitatory, but this time our postganglionic neuron's unmyelinated axon goes out the gray ramus. And from there, it can go out to the dorsal ramus and the ventral ramus to affect the skin. And someone mentioned about a U-turn. It is kind of like a U-turn, but like I said, notice it's also like a 360 because you go past the exit, come down in, and then come back out to where you were going before. So it is indeed a U-turn. In fact, it's practically a 360 if you're going out to the skin. So hopefully seeing the words again, hopefully seeing this illustration helps you to understand that. And again, this picture is not from your textbook. Whoops. There, where are we going? This picture is not from your textbook, but I like it because it shows the paired sides. This is the picture from your textbook, and it shows the same thing. Lateral gray horn for that preganglionic neuron, ventral root, spinal nerve, white ramus, synapsing in the chain, and then coming out to go to the skin. The reason it can't go back straight out is because these 2 communicantes are different. One is the white ramus, one is the gray ramus. The white ramus is made up of myelinated axons. The gray ramus is made of unmyelinated axons, right? So it's like trying to drive down, you're trying to drive down J Street in downtown Sacramento. You can only go one way. If you want to turn around and go back, you have to go over a street and then come down H instead. Right? So we've got a bunch of one-way streets here. These 2 are one-way streets. Oh, and the reason it can't go straight out is, remember, our autonomic pathway requires 2 neurons, right? We can't go straight from the central nervous system to the effector. In our sympathetic pathway, we need 2 neurons. Now, why do we need 2 neurons? Especially since if you think about it, this one's always excitatory and always stimulates the other one. So why not just hardwire them together? Why not just have 1 neuron? Why is one of those questions we can't ask in anatomy and physiology. Again, the engineer who designed all this did not leave us her footnotes, so we don't know why, but that's the way it is. For whatever reason, our autonomic pathways take 2 neurons. So that's why it can't go straight. And again, your book does a nice job of showing that ascension, but notice you're still synapsing, you're still going out the gray ramus, you're still going to the skin. But remember also, we don't have to synapse in a chain ganglion. Instead, we can synapse in a collateral ganglion. Again, the path starts the same way. Preganglionic neuron, whose cell body is located in the lateral gray horn. Myelinated axon comes out the ventral root, through the spinal nerve to the white ramus, and from the white ramus to the chain ganglion. But remember, we do not synapse in the chain ganglion. Instead, we pass right through the chain ganglia, and our preganglionic myelinated axon forms the splanchnic nerve. That splanchnic nerve travels to our collateral or prevertebral ganglion, and there it's going to synapse. And again, you have 3 choices. It can either be the celiac, it can be the superior mesenteric, or the inferior mesenteric. And of course, which one you synapse in determines which effector you're going to. Right? Stomach, spleen, liver, gallbladder, right? Pancreas, all of those are celiac. Small intestine, large intestine is the superior mesenteric. Rectum, anus, urinary system, reproductive system, inferior mesenteric. All right. And then, of course, that unmyelinated postganglionic axon leaves, and again, it leaves just as a sympathetic nerve and innervates the abdominal pelvic region. Here again is that pretty picture that's not from your textbook. And again, I like it because, again, it shows both sides. Both sides, the same thing happens. Our preganglionic neuron comes out, ventral root, spinal nerve, white ramus, through the chain without synapsing, out the splanchnic nerve to a collateral ganglion. And the exact same thing on the other side, because remember, our chain ganglion are paired, our collateral ganglia are not. There it synapses, and from there, again, those postganglionic fibers, which just form sympathetic nerves, go to the stomach, the gallbladder, the spleen, the small intestine, the large intestine, the bladder, whatever it is, some organ below the diaphragm. Again, here's the picture from your textbook. It only shows one side, but again, you got to love those artists. Notice, and again, I will say in fairness, they give us the pathway, lateral gray horn, ventral root, spinal nerve, white ramus, through the chain, into a splanchnic nerve. But notice, that splanchnic nerve leads to a collateral ganglia, and they do say such as the celiac. But then they have that postganglionic neuron innervating the intestine. Now, for starters, they didn't say small or large intestine, which is a mistake. But more importantly, does the celiac ganglion innervate any intestine? Yes or no? Does the celiac ganglion innervate any of our intestines? No. And again, in fairness, they say such as, in fairness, they do say, for example, e.g., but it would have been nice if they would have at least matched those two together. But ironically, they did not. But it still shows the pathway. And then again, the fourth pathway, if we're thinking in terms of where the synapse takes place, the synapse could take place in the suprarenal medulla. And again, remember, suprarenal is just a fancy way of saying the adrenal gland. The adrenal gland sits on top of the kidney, so it is often referred to as the suprarenal gland. And again, we want to go to its medulla. It's a triangular shaped organ with a chewy nougat center and a candy-coated shell. And the outer part is called the cortex, and the inner part is called the medulla. So our goal is to get here to the medulla of the adrenal gland. And as we keep saying, it all starts the same way. Ventral horn, lateral horn, ventral root, spinal nerve, white ramus to the chain ganglia and do not synapse. Out the splanchnic nerve, and again, we want to make sure we emphasize that for our pathway. Out the splanchnic nerve. To not just any collateral ganglia, but specifically the celiac ganglia. And again, do not synapse in the celiac ganglion. Instead, pass all the way through the celiac ganglion, all the way to the adrenal gland, and finally synapse in the medulla, where our postganglionic neuron releases our hormones into the circulation. That postganglionic neuron produces about 80% epinephrine, or adrenaline, and about 20% norepinephrine, or noradrenaline. And for our purposes, chemically, these two are almost identical to each other. And for every function that we will look at, their functions are the same. Right? When you get to graduate school, you'll get to learn about the subtle differences between adrenaline and noradrenaline. But for now, they're pretty much interchangeable chemicals that do the exact same thing for everything that we care about. And we've done it with the words, so of course we have to do it with the pretty pictures. And as I mentioned again, this picture that's not from your textbook does the best job of showing this. Notice we still have two neurons, a preganglionic that starts in the lateral gray horn, ventral root, spinal nerve, white ramus, through the chain, splanchnic nerve, to the celiac ganglion, no synapse, out the celiac ganglion, to the medulla of the adrenal gland, synapse on the postganglionic neuron, and that postganglionic neuron releases hormones into the blood. And those are your four pathways. Again, So great question. What you're talking about is this part right here. And again, what this really has to do, as we talked about, is some of the elaborateness. There are most of the pathways do indeed go through the inferior mesenteric. Remember, the parasympathetic don't go to any ganglion. Again, one of the things you got to remember is everything we talk about is a little bit more complicated. There are some sacral ones that have some minor input, but this is the major input. So the same way that there is some minor input from the superior cervical to the heart, there is some, and notice they're still not direct. There are some synapses in the chain. There are some minor inputs to the reproductive organs that don't pass through the inferior mesenteric ganglion, but most of them do. So what I would say is the sky's blue answer is that these things are innervated by the inferior mesenteric. But again, remember, our sympathetic is big and global and messy. And there's lots of different collateral routes to get to the places they want to go. And so there are some minor inputs that avoid that ganglion, but it isn't something that we have to worry about. All right. And so again, yep. So then again, if you think of what can happen inside of a chain ganglia, as we talked about, remember when you come to that chain, you can synapse right away. You can ascend or descend. You can continue through and synapse in a collateral ganglia, or remember there's actually a fourth thing you can do. You can also pass through it, and that's the one they didn't draw on this picture. You can continue on through without synapsing and then go to the adrenal gland. So there's lots of ways to look at this information and think about this information, make sense of this information, right? And that's kind of what the next couple slides are about. Although this one I want to talk about, but I think, again, this is a good stopping point. Let's go ahead and take our break, and then we'll come back. This is the last real big piece that we want to talk about, that I want to discuss. And then from there, we can move forward to the last little bit of information we need to talk about for today. Let's go ahead and clear that. I'll go back to this. This is a good stopping point. So let's go ahead and take our next break. My watch says 2:06, so we'll come back at 2:17. And restart and record. So pause. We are down to our final stretch. So again, as I mentioned, to completely beat the dead horse, there are just a couple more ways that I want you to think about this. Another way to think about this, and again, remember, anytime we're thinking about these things, and I say they're good ways, those are always things that tend to make decent essay questions. You know what? I'm not going to do this. Let's do this instead. I'm going to cheat and use our picture here, because this picture from your textbook is mostly good, but remember, it needs an additional arm. There we go. One of the important things to remember is that there are basically 6 structures that either feed into or out of the chain ganglion. Right? Anytime you're giving driving directions, there's always that big intersection where a lot of things pass through or a lot of things go. And so understanding and appreciating what they are and where they go and what they're made out of is one of the ways that we can get a better understanding of the pathway. So what we can do here is number them. So let's do this. We'll make this one number 1, and this one number 2, and this one number 3. Oops, I guess number 3. There you go. Does that show up there? Excellent. This one will make number 4, and this one will make 5, and this one we will make 6. So that we can identify these 6 possible structures feeding into and out of them. And for each of these structures, we need to know basically a bunch of pieces of information. First, we need to know the direction it goes. Well, first, actually, I guess first thing we need to know is their name. Second thing we need to know is the direction axons travel in that structure. And again, basically, there's 2 answers to that. Do they travel in or do they travel out of the chain ganglion? Third thing we need to know is the type of axon. Is it preganglionic or postganglionic? And then, of course, depending on the type of neuron, will tell us if it's myelinated or unmyelinated. And then the last thing we need to know is where it's going. If it's coming in, if it's going out, we need to know where it goes, or we need to know where it came from if it's coming in. So again, depending on if it's coming in or out, we need to know where is it going or where is it coming to, or where is it going from. So let's do that. Starting first with number 1, and number 1 we picked on purpose because it's easy, because we can see that there is an axon coming into it. So because that one's coming in, one of the structures, and again, technically it could be any of the 6, but since we have this drawing here, the first one clearly has an axon coming in it, so that first one is our white ramus. And the white ramus, of course, is its name. We know axons travel into the chain from the into the chain, and we know they're coming from, so they're into the chain, let's write that, and they're coming from the lateral gray horn. All right, this is the axon of our preganglionic neuron. All right, so it's the preganglionic axon that forms it. And of course, because it's the preganglionic axon and because it is the white ramus, we know it is myelinated. All right. That's one. And we see the axon coming in. My guess then is that 2 must be the gray ramus. Does the gray ramus come into the chain or out of the chain? Out of the chain. Excellent. Right? And it's going out of the chain to go where? Where are these axons going out of the chain going? Excellent. I like that. To the ventral ramus is absolutely correct. Dorsal ramus would have been good as well. Or if you think about it, we could have also said the effector it was going to as well. So skin probably would have been a good answer there as well. But I like ventral ramus. That is great. Awesome. Is this a preganglionic or a postganglionic axon that forms the gray ramus? Excellent, postganglionic. And that, of course, means myelinated or unmyelinated. Excellent. It is postganglionic and unmyelinated. Excellent. Let's jump to 4, because again, that shows us, it cheats a little bit in this picture and shows us what it is. So 4 is, and so we'll cheat here. It is clearly going out of the chain, right? We can see we need to know where it's going to. We'll get to that part in a second. We can clearly see it's a postganglionic axon. And so, as we already mentioned, that means it's unmyelinated, right? So we have all of that. So really, the only 2 pieces of information we're lacking is what this structure is called and where is it going. Anybody know which this structure must be called that's made up of the postganglionic axons, unmyelinated leaving our chain? Not a bad guess. Now, remember the gray ramus, it goes to it, and the ventral root comes to the white ramus. Now, this, remember, is just our sympathetic nerve. And if it's a sympathetic nerve, where is it going to? Where are these axons going? Yep, these are going to the effector, absolutely. Although, again, at this point, we can be a little bit more specific. You're right. I like that, though. I like that. This is going to effectors, and here we can be more specific. Effectors above the diaphragm. There you go. To effectors above the diaphragm. So that would be the head, the heart, the lungs, something like that. Excellent. Means we skipped 1, 3. 3 is our splanchnic nerve. Now, from what I remember, the splanchnic nerve's coming into the chain or out of the chain? Excellent, out of the chain. And where are they going to? Where did our splanchnic nerves go? Anyone remember? I'll give you this part. It is going to go to effectors below the diaphragm ultimately. But if you think about it, so you're absolutely correct about that. However, in this case, the splanchnic nerve goes out of the chain to the collateral ganglion, to synapse, right? Because it has to synapse in that collateral ganglia. And you're right. Once it synapses in that collateral ganglia, it will go to an effector below the diaphragm. But first, it has to synapse in that collateral ganglia. And if it's going to the collateral ganglia to synapse, is it a preganglionic or postganglionic neuron? Pre, excellent. ganglionic neuron. And if it's preganglionic, of course, it is myelinated. Perfect. And really, that only leaves us 5 and 6. And notice I'm putting 5 and 6 together because 5 and 6 are the ascending. And I guess we could put them in order, but we'll put them together. 5 is, actually, I guess 5 is the descending, the way I've numbered them. Oops. And 6 would be the ascending, I guess, as we have labeled them here. Chains. Oops, wow, that doesn't even close. Chains of our chain ganglion. They are going to be made of axons. And here's the only tricky part with this one. If you think about it, let's start here. This axon coming in could have ascended to go up or could have descended to go down. But at the same time, a chain ganglion from below could have an axon that is ascending as well. So as it turns out, in this chain, things go, and that color is horrible, so let's switch to the dark red. Things can go in both directions. With our chains, we can have axons that both go into, well, again, the axons don't, but we can have both types. We can have axons that go into and others out of the chain. So basically, axons can go in both directions. Of course, remember, these are still axons from before they synapse. So they're still all going to be preganglionic. And so, and so are they going to be myelinated or unmyelinated? Preganglionic neurons, axons are myelinated. Excellent. There you go. So again, we have our major hub, our highway and byway, where everything comes into and out of, right? This is kind of like the corner of Madison and Auburn, right? There's a bunch of traffic going all sorts of different directions. But if you can tell what's going what direction, you have a good, strong, sound understanding of what is going on. So those are our 6 pathways into or out of our chain ganglion. All right. So again and again, I didn't write all that out, but again, you should be able to identify the 6 pathways, know the direction axons travel, know whether they're a pre or post ganglionic axon, which of course also tells you whether they're myelinated or unmyelinated. And then you should be able to tell me either where they're going or where they came from. And that also sounds like a really good essay question. All right, the last of these questions here, again, are just different pictures that, again, emphasize this. I totally want to beat this dead horse because I totally want to understand it, and because I know it can be a bit confusing. I'm hoping by showing you lots of ways to think about this helps something to click to make sense. Notice this is a great illustration from your textbook, which again kind of shows, all right, where are the neurons' cell bodies located? All preganglionic neuron cell bodies are located in the lateral gray horn of T1 through L2. All postganglionic neurons are located in one of 3 locations, either inside a chain ganglion, inside a collateral ganglion, or inside the medulla of the adrenal gland. And if this is where our postganglionic neuron is located, then what effector is it going to influence? Notice if you're in a chain, you're either going to the head or the thoracic cavity or the walls of the body. If you're in a collateral ganglion, you're going to an organ below the diaphragm. And if you're in that adrenal gland medulla, the medulla of the adrenal gland, you're releasing your hormone into the blood and you're affecting everything. Lastly, we can think of it in terms of those nerves, right? What is the ultimate end of the pathway? We have spinal nerves, right, coming out the gray ramus. So that's the dorsal ramus and ventral ramus. They're the ones that go to the skin, the sweat glands, the erector pili muscles, the blood vessels, right, to those dermatomes. If you come out of a sympathetic nerve from the chain ganglion, you're going to the head, the heart, the lungs, the stuff inside above the diaphragm. Splanchnic nerves go to the prevertebral ganglia, and as someone mentioned, those go to the organs below the diaphragm, gastrointestinal tract, the stomach, rectum, urinary system, reproductive system, or remember that one splanchnic nerve till the celiac trunk goes also to the adrenal gland as well. So again, I've tried to give you as many ways to digest this information and make sense of it so that you can understand and describe these pathways, all associated with this scary octopus. That again makes sense. And again, once more, the more we look at this, the more beautiful we see this is. Notice the cervical is going to the head, right? Here we've got, well, actually, this should be going to the heart and lungs from the cervical here, so that's not ideal. But we have them coming off the chain, ganglia going to these structures. Collateral ganglia are going to the stomach, the pancreas, the liver, the gallbladder. And notice we've got a path that goes all the way directly to the adrenal gland as well. So these are, even a simplistic octopus like this kind of starts to show us some of the stuff that we need to know. All right. Now, last little bit, that was the heavy lifting, so now it's kind of easy riding from here on out. Now, the good news is a lot of the stuff we're going to talk about is really not new information. We may be adding some vocabulary to things we already know, but at this point, there's not a lot of new information. Now, as we've said, nerves form elaborate networks that we call plexes. We saw the spinal plexes, and there were 4 of those, and it talked about and identified those. Those you need to know the names and the roots that make them. There are sympathetic and parasympathetic plexes as well. There are about a dozen of them. You don't need to know them. And even though I've listed some of them here, you don't have to know all of these. In fact, there's only really one I'm going to talk about. But not surprisingly, as we talked about, they're often associated with blood vessels. Here we have a pretty picture that shows this. I love this picture because this picture does a great job of showing us our chain ganglion, how they come down and come up the other side. Here we see some of those elaborate networks of nerves, and more importantly, here in front we see our celiac ganglion, our superior mesenteric ganglion, and our inferior mesenteric ganglion. And we see that some of these ganglion are associated with elaborate networks of nerves. In fact, both our celiac and our superior mesenteric ganglion are associated, and here we can see it a little bit better. We get the pesky person out of the way. We can see those chain ganglion coming up and coming down. We can see the collateral ganglia located here. And what I want to show you is this elaborate plexus right here. This elaborate network of nerves that includes the celiac ganglion and the superior mesenteric ganglion here form a plexus known as the celiac plexus. Or maybe you know it by its other name. The other name for the celiac plexus is the solar plexus. And there's one other structure that I want to show you on this, although it doesn't really show it on this illustration. But while here we see all of the sympathetic and parasympathetic nerves, as it turns out, there is another nerve that travels down this merry path and comes and becomes a part of this celiac plexus. And that other nerve that comes down here is the phrenic nerve. Remind me again why that sounds familiar. What does our phrenic nerve innervate and control? The diaphragm. It controls the diaphragm. You may have never thought of the phrenic nerve before, or even the celiac or solar plexus, but you and a buddy could have been sitting there talking on the side of a soccer field when, unbeknownst to you, someone kicks a soccer ball really hard. And without you being aware of it, it happens to hit you in the stomach, not really in the stomach. It hits you in the abdominal region, right above your umbilical region, right above your belly button, or more specifically, right below your xiphoid process of your ribs, right? And what happens is that increase in pressure, that sudden increase in pressure here below the ribs disrupts the activity of the celiac plexus. So yeah, that disrupts the activities of some of the autonomic functions on the inside. But what you notice the most is that it disrupts the activity of your phrenic nerve. And suddenly, you're not so able to control your diaphragm anymore, right? If you can't control your diaphragm, do you die? No, but you feel like you're going to, right? Luckily, we have accessory muscles that can help to elevate the ribs and help to force air out. So we can raise our hands up in the air to make it a little bit, but you're not going to die, but you're sure as heck going to feel like it when you can't control your diaphragm because you have a lot harder time getting a deep breath, right? And so there you are panting and in pain. And what do we often refer to that as? There you go, getting the wind knocked out of you. Right? When you get the wind knocked out of you, that's exactly what's happening. That pressure from that unexpected blow that you weren't able to tighten up your abdominal muscles and protect yourself from has disrupted the activity of your celiac plexus, has disrupted the activity of your phrenic nerve, and now you, for a few minutes, have trouble controlling your diaphragm. And you get the wind knocked out of you. So that's the one plexus I wanted to point out because, like I said, that's the one that most people at this point in their life have had the wind knocked out of you. And if you haven't, then I encourage you to definitely go and get that done. No, I'm just kidding. Don't do that. Great question. Yes, absolutely. The hiccups are also most hiccups. I guess there are some that are not, but often hiccups are associated with a disruption in the activity of the phrenic nerve. That is correct. The activity gets disrupted. So there are several ways that you can reestablish that. Change the subtlest is to change your breathing habits. Breathe slowly and steadily. And every time you hiccup, then what you do is you change the position of your diaphragm. So if you were inhaling when you hiccuped, instantly start exhaling. If you're exhaling when you hiccup, instantly start inhaling. A few moments of that can do it, but there are plenty of tricks that people try. Drinking a large glass of water upside down is something that people do. Scaring people is another way where basically that's kind of like the the shocking of the heart to bring it back. You're basically disrupting the neural activity to be able to do it. Although you can also do what my daughter's favorite way of doing it is. One of the interesting things about the phrenic nerve, because remember, the phrenic nerve comes way up off of the cervical plexus. And one of the things that happens to run past it in the pathways is the facial nerve and some of the nerves associated with taste. So one of the things that can sometimes disrupt the activity of the phrenic nerve is to overwhelm it. She tells, if I give her $1, she hiccups again and it works. All right, there you go. But what my daughter likes to do instead is to disrupt the activity of the phrenic nerve. And so sometimes eating something really sweet. And so basically, if you take a spoonful of sugar and put a spoonful of sugar on your tongue, sometimes the size of that large neural signal to your brain telling you that you perceive something sweet can disrupt the neural flow to the diaphragm and can actually reset it as well. So anytime she starts hiccuping, she goes begging for a spoonful of sugar. So there you go. So yeah, everybody's got their trick to be able to help you to do it. But absolutely, that phrenic nerve, disruption of its activity, disruption of the diaphragm is associated with that. And so is getting the wind knocked out of you. All righty. So that is it for the nerves. The last little bit, and we talked about this a little bit before, are the neurotransmitters and the receptors, right? Remember, one of the things that we talked about is that when talking about communication at a synapse, where's my drawing stuff? We have a target cell. And that target cell is often a neuron, but whatever that target cell is. And then we have a neuron. There are two things that we have to take into consideration when understanding the connection here. The first is the neurotransmitter that is released, like, for instance, acetylcholine. But remember, we said acetylcholine wasn't by itself excitatory or inhibitory. What determines whether it is excitatory or inhibitory is the type of receptor that it communicates with. As we talked about it, and heck, I'll cheat and make 2 cells, even though I made my first cell so big. Remember, one of the things we talked about is acetylcholine on some cells can be excitatory, and on other cells, that same acetylcholine can be inhibitory. So if the target cell, for instance, was the heart versus, or let's be more specific, cardiac muscle versus skeletal muscle. Remember, on skeletal muscle, that acetylcholine, when it binds, is going to be excitatory. And we said the reason it is excitatory is what that acetylcholine actually does is it opens a sodium channel. Really, it opens a cation channel that, as we know, where it's both sodium and potassium flow. But as we talked about, it is mostly the influx of sodium that causes that excitation, causes that depolarization, causes the muscle cell to be stimulated. However, there is a second type of acetylcholine channel. This one, as we talked about on our cardiac muscle, actually opens chloride channels. And if we open chloride channels, we of course know that chloride is going to enter into the cell. But whereas sodium entering is excitatory, chloride entering is inhibitory. And our heart beats slower as a result of it, right? There's absolutely no new information here on the board. This is all stuff that we've already talked about before. That how a neurotransmitter affects a cell is not just the neurotransmitter, but also the receptor. And so really, the only new thing we need to add to this are some fancy names, because what do anatomists love? They love their fancy names. Starting first with this neuron right here. This is a neuron that releases acetylcholine as its neurotransmitter. I don't have any idea why I capitalized the H, but I guess I can still hear. Releases acetylcholine as its neurotransmitter. All right, so it releases acetylcholine. So this neuron, by definition, we call a cholinergic neuron. Cholinergic neuron just means a neuron that releases acetylcholine. And if you think about it, we've run into a lot of these. Remember, we have our 2 neurons in our autonomic pathways. Our 2 neurons in our autonomic pathways involve our preganglionic neurons, both in the sympathetic, and our preganglionic parasympathetic neurons. They all release acetylcholine. Remember, all parasympathetic postganglionic neurons release acetylcholine. I think at this point I'm going to have to erase my picture. So clear all drawings. Remember, we said most sympathetic release norepinephrine, but we did say a few do release acetylcholine. In particular, the ones that release acetylcholine are the ones associated with our sweat glands and the ones associated with the blood vessels of our skeletal muscle and our brain. And then, of course, the very first neuron we ever learned about was that somatic motor neuron that communicates with our skeletal muscle. And we know that one releases acetylcholine as well. So these are all neurons at some part during the class that we have talked about. and we've said all of them release acetylcholine, and so all of them are cholinergic neurons. A cholinergic neuron just means it releases acetylcholine. And here are some of the examples we've talked about in the class. Again, really no new information here, except maybe we're specifying which sympathetic ones release acetylcholine. But we've just given them a label. We have referred to them as cholinergic neurons. The other part that isn't terribly new, because I already kind of hinted at it with the previous picture. I wonder if I can undo my picture now. Ah, yay, it's still there. Perfect. Therefore, a receptor that acetylcholine binds to gets the fancy name cholinergic receptor. And as I already hinted at, there are two different cholinergic receptors. For right now, we'll call them A and we'll call them B. That would be awesome if that was the names they use, but of course we know anatomists hate us, so they wouldn't use terms like A and B. But we do need a way to tell them apart. Now, if I have a third cell, and let's go ahead and draw a third cell here, and give it a mystery receptor that we don't know which it is. We don't know if it's green or we don't know if it's brown. We don't know if it's A or we don't know if it's B. If we release acetylcholine and it binds to that channel and opens the channel, do we know whether it's A or B? Well, does acetylcholine bind to A? Yes, and opens it. Does acetylcholine bind to B and open it? Yes, it does. So if acetylcholine binds to it, that doesn't tell us anything. So we can't use acetylcholine other than to tell us it's a cholinergic receptor, and that doesn't help. Now, as it turns out, these channels are similar, but they're not identical. There are some subtle differences. And where we see the differences are actually in the toxins that can bind to them. It turns out that, as we've talked about, we know there are toxins that can bind to acetylcholine receptors. We talked about curare being able to bind to acetylcholine receptors. We talked about tetanus being able to bind to these acetylcholine receptors. So we know toxins can bind to acetylcholine receptors. Well, it turns out receptor A has a special toxin that can bind to it, but cannot bind to receptor B. And it turns out that toxin is nicotine. Where do you get nicotine from? Cigarettes. There you go, from smoking. There you go. So it turns out nicotine binds to receptor A, but does not bind to receptor B. So if I take this nicotine and release it here, and it binds and it opens this channel, now I know it's channel A. And so what I did is I said, well, since nicotine opens channel A, well, I didn't, but this is what an anatomist did. Since nicotine opens channel A, instead of calling it channel A, I will call it a nicotinic receptor. Oh, that's way off. There you go. Nicotinic receptor. So A isn't its name. A was a stupid name anyway, so we'll get rid of that. And instead, we're going to give it a fancy name. It's a nicotinic receptor because the toxin nicotine will bind to it. Turns out there is a toxin that binds to B as well. In fact, as I said, my former student's boyfriend knows it well. It is a toxin called muscarine. Muscarine is the toxin that you find in wild mushrooms, deadly wild mushrooms. That's the one that we talked about, stimulates all those parasympathetic responses, the constriction of the airways. Nope, not the sodium channel. That cation channel, you were right. The cation channel that is on our skeletal muscle is a nicotinic receptor. Yes. That muscarine that causes the heart rate to decrease, the pupils to decrease, the respiration to decrease, all those things we talked about is because of a poison, a toxin that's in those mushrooms called muscarine. And it turns out muscarine binds to B, and so we call it a muscarinic receptor, and that way we can get rid of that B as a silly name after all. So now, if I release nicotine on this and it opens, I know it's A. If I release muscarine and it opens, I know it's B. By using the toxin, I can tell them apart, so I identify them by their toxin. And so as it turns out, there are two types of cholinergic receptors. The first is nicotinic, stimulated by nicotine. These, like in our skeletal muscle, so you're absolutely right, that cation channel on our skeletal muscle is a nicotinic receptor. It is always excitatory. So if you think about all the places we said where acetylcholine is always excitatory, the neuromuscular junction, where our preganglionic neuron communicates with our postganglionic neuron. All of those places where it's always excitatory, it uses that nicotinic receptor. Whereas the muscarinic receptors can be either excitatory or inhibitory. And so, for instance, this is the one that's found on the heart. This is the one that's found on our sweat glands. These are the ones that do that, like I said, cause those effects from that poisonous mushroom. Because if you know the kind of receptor, you can know what the effect will be. So if I tell you that a skeletal muscle has a nicotinic receptor and I release acetylcholine on it, is it going to stimulate the muscle to contract, or is it going to cause that muscle to relax? Yeah, it's going to cause it to contract. So we know that, right? Whereas, like we said, you instead put acetylcholine on the heart and it slows it down. And like I said, it inhibits it. So again, it can be confusing for some people as to why is it that if I put acetylcholine on one thing, it's excitatory, and if I put it on another thing, it's inhibitory. And the answer to that is they have different receptors. It's not the acetylcholine that's different, it's the receptors that are different. And those receptors do different things. So understanding what those different types of receptors can do is going to be important. And not only is that important for cholinergic receptors, but it's also going to be important for adrenergic receptors. And again, this may be where it helps to make a little bit more sense in why you will see why this is relevant. It turns out, in the same way, and I'm going to cheat. Can I bring back my picture? Yes, excellent. So I need to cheat now. Get rid of that, get rid of that, get rid of that, get rid of that, and get rid of that, and 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 that. Excellent. Once again, I have a neuron, and this neuron releases norepinephrine. And a neuron that releases norepinephrine, we give a fancy name to, and that fancy name is an adrenergic neuron. And of course, as we have seen, there's really only one adrenergic neuron that we have talked about in this class. Move that up and out of the way. Neurotransmitter released. Most of our sympathetic postganglionic neurons. Now, we may release, let's get rid of this stuff too. Norepinephrine. But as it turns out, there are two major classes of adrenergic receptors. And once again, as a starting point, we can use their names as A and B. But again, A and B is a stupid name for them. We wouldn't want to use A and B. However, if we use the Greek versions of it, it sounds way more cool. And so that is exactly what our anatomists did. The two main classes are alpha receptors, A receptors, and beta receptors, B receptors. So when we use the Greek, it's way more fancy than just A and B. But both alpha receptors and beta receptors have specific types. In fact, our alpha has 2 specific types, and our beta receptors actually have 3 specific types. So let's take a look at those. Starting first with the alpha. Again, understanding where these are located helps us to understand how to manipulate this system. Alpha A, which are our most common type of adrenergic receptors, are the ones that are found on our smooth muscle of our blood vessels and our digestive system and places like that. And they are always excitatory. So again, when we have our stress response, that stress response sends a signal to our blood vessels of our digestive system. Those blood vessels are stimulated and they constrict. So less blood goes to our digestive system, so we can send blood to other places instead. At the same time, in areas like our salivary gland, we want to decrease the metabolic activity. If we can decrease that metabolic activity, right, then again, we can use energy elsewhere. So while the blood vessels in your digestive system are constricting, they're more active, constricting, so less blood goes there. If we decrease the metabolism of our salivary glands, we produce less saliva, or we produce less mucus, right? If we can stimulate these alpha-2 receptors in our nasal cavity when we have a cold, we can stop our nose from running. And that's what a lot of those, runny nose medicines that you take do. They stimulate the alpha-2 receptors to decrease mucus production, so your nose isn't constantly running. But here's the one that most of you are probably familiar with. You've probably never heard the terms cholinergic and adrenergic before, but one of the things you probably know, especially because it's more relevant now with the coronavirus, maybe you know someone who's had a heart surgery or a heart attack or has bad high blood pressure, right? When you just had heart surgery and you come home, one of the things that might be important is to be able to, exercise. And part of that exercise could be carrying your groceries up 2 flights of stairs to get to your apartment. However, as we know, when you carry your groceries up 2 flights of stairs, you're exerting yourself. And when you're exerting yourself, you stimulate your sympathetic nervous system. And one of the things that happens is your heart beats faster and your heart beats stronger. And if you've just had heart surgery, do you necessarily want your heart beating stronger and your heart beating faster? No. So if you've just had heart surgery, one of the things that the doctor may put you on is a certain type of medication. And that certain type of medication is referred to as what? There you go, beta blockers. And that's exactly what we're talking about here. Beta blockers actually block the beta 1 adrenergic receptors. These are the ones that are found on your heart, so that when your body releases adrenaline or your body releases norepinephrine, right, those bind to those beta 1 receptors on your heart, and your heart beats stronger and your heart beats faster, which is a really good thing, except when it's not. And if you don't want your heart beating stronger and your heart beating faster, you can take what essentially is a toxin that blocks those beta 1 receptors. And that's what a beta blocker is. Those beta blockers block the beta 1 receptors, decreasing the effect that your sympathetic nervous system has on your heart. And now your heart's not going to beat stronger, your heart's not going to beat faster, and it's going to be much more safe, much more healthy. Beta 1 is the one that I like, or beta 2. Actually, both of these are the ones that I really like, because this is the reason why I carry my EpiPen with me anywhere I go with my daughter. Because if she goes into anaphylactic shock, because I'm sure, as I've mentioned numerous times, she is a severe peanut and tree nut allergy. And if she were to accidentally eat the wrong thing or come in contact with the wrong thing, she could go into anaphylactic shock, where her blood pressure drops. No, thankfully, we have never had to use the pen. She's never had anaphylaxis, knock on wood. We have not had to use it yet, and hopefully never will. But again, knock on wood. With people who have anaphylactic shock, you have a decrease in heart rate. And like I said, you get a thickening of the mucous membrane in the walls. Well, you give them that shot of adrenaline, and that adrenaline runs to the beta 1 receptors, stimulating the heart to beat stronger and beat faster, bring the blood pressure back up. It also goes to the beta 2 receptors, which are in the smooth muscle of your lungs, opening up your airways, helping them to get more air. Right, so being able to stimulate those with adrenaline, with our, adrenergic neurotransmitter, epinephrine and norepinephrine, are important. However, as important as these are, especially now that we're all at home eating 24 hours a day, prior to this current pandemic we've been dealing with, there was a tremendous amount of energy and money going into research of beta 3 receptors. Turns out beta 3 receptors are excitatory receptors that are found on our adipose tissue. And they basically do a couple things. They help us to metabolize fat to maintain our body temperature. But the also thing they do is they help to encourage cells to be more likely to break down fat to metabolize energy for ATP than glucose or amino acids. So again, think of the implications of that. One of the things that you can do if you want to have that beautiful body, which as we all know is the only thing that matters, you could go to the gym and work out constantly and have a really high metabolism and eat really low amounts of food and eat really lean foods so that your body basically eventually starts breaking down those fats because you've got more calories going out than coming in, and you can maintain that beautiful figure of yours. However, what if instead you could sit on a couch, take a pill, and that pill here, sure, might make you sweat a little bit, may make you a little warm on the inside. But if it actually, that pill stimulated those special beta 3 receptors that just cause the lipids to just start melting away from your body and being released into your blood so that it's no longer inside that subcutaneous fat. And so we could all be sitting in our chairs at home, right, drinking heavily from the pandemic and all look beautiful when we get done. Right now, obviously, there's more to it than that. But again, a way to get the body to more efficiently metabolize fat is a magic pill if we could figure out how to do that. And so there are a lot of people who are looking at studying these beta 3 receptors. So yeah, knowing these receptors and understanding what they do is something that can be useful. So yes, the question is obviously when you release adrenaline into the blood, adrenaline affects all of these, right? Because again, all of these are receptors that respond to adrenaline, epinephrine and norepinephrine. But as we kind of hinted at, again, really from the beginning of this class, we can also affect these things with toxins as well, with other chemicals. And I keep using the term toxins, but notice some of these things we talked about are medicines. And again, this is not a new concept. What is new is if we give it a fancy name. So first, you have a nice table that talks about the different types of receptors, alpha, beta, and the adrenergic, the cholinergic ones. Make sure you know that. But here's the last little bit. And again, it's not really new information for us. We are just now given a fancy name to it. Remember, way back in the skeletal muscle system, we talked about tetanus. And we talked about how tetanus binds to what we now know is a nicotinic receptor. And it opens it, causing all of that sodium to come in and causing that rigid paralysis. That's because tetanus is what we call a receptor agonist. An agonist binds and activates the channel. Do we need to know all the receptors are? Yes, you need to know all the receptors. So all 7 different types of receptors, the 2 cholinergic, the 5 adrenergic. Yes, you need to know all of them, where they're typically found, what their typical effects are. Yes, you need to know all that. Again, you need to know everything we talk about in class. I've got 5 slides on this stuff, so obviously it's stuff that's important. Receptor agonists bind to and activate the receptor. So they basically mimic neurotransmitter. Tetanus was one of those that we talked about. But here's the other one, as I mentioned, that phenylephrine binds to A1. That's the one that we said constricts the blood vessels going to your digestive system or going to the mucous membrane of your nose, causing that decrease in mucus production. Right? Dilates the pupils. It increases blood pressure. It acts as a decongestant. It makes you feel better when you're sick. We talked about nicotine, right? Think back to your first cigarette, right? Third grade, you bum one out of your dad's pack, ran to the backyard to smoke it, right? You got all shaky, got a little nauseous, everything that went along with that. And unfortunately for some of you, it was pretty addicting, right? Remember, one of the things we talked about is dopamine plays a role in some of the pleasure sensations, and in our insula plays a role in addiction. So it can stimulate that addiction. It releases epinephrine, makes you alert, makes you awake, makes you feel good, right? Those cigarettes are powerful, powerful addictive drugs, right? Well, actually, it's the nicotine in the cigarette, right? Which is what makes things like those vaping things even worse, right? Because they could be just as bad as the cigarette, but they look so cool. Conversely, as we also talked about, some things bind and block the receptors. These things are called receptor antagonists. An example of receptor antagonist that we already talked about is curare. Remember, it binds and blocks the nicotinic receptor on our skeletal muscle so that we get that flaccid paralysis. My favorite example of this, though, is atropine. All right. Back in ancient times, you were allowed to be within 6 feet of people. And one of those people you might occasionally have to be in close proximity with would be an optometrist. Optometrists in general are horrible, horrible individuals. They're just like practically the worst people ever, right? And one of the things that makes them so horrible is they only have eye exams on noon on sunny days. So the only time they have an appointment is at noon o'clock on a sunny day. So they get you there in a nice, bright, sunny day into your dark room, stick you in the chair. And then, because they're too lazy to move a little bit to the left or a little bit to the right, they put a drop into your eye, dilating your eyeballs, right? Your iris. That drop they put in your eye is something similar to atropine. It's not actually atropine, because atropine would actually last for, 6-8 hours. Whereas the drop they put only lasts for like 3 or 4, which is not nearly as bad, right? Horrible, horrible people. And what that does is it blocks the parasympathetic input into your iris. Remember, we talked about there's that autonomic tone. There is always sympathetic and parasympathetic going in. We block the parasympathetic, and remember, parasympathetic is what constricts the pupil. Then only sympathetic works, and so your pupil dilates. And when they shine that blazing bright light into your eye, your parasympathetic can't close it, and allows them to get a really good look at your retina. Of course, once they're done with that, like I said, it lasts for hours. So then they kick you out of their office, and then you're out there in the blazing sun, and all they've given you are those crappy little plastic wraparound sunglasses, right? Horrible people, like I said, horrible, horrible people. All right, but the point is that drop they put in your eye is a parasympathetic antagonist. It binds and blocks to it, dilates the eye. It also decreases the mucus production, so you get a dry nose from that and really gross yellow-looking boogers. Like I said, the horrible, horrible people. And again, that other classic example we talked about, the beta blockers, right? They block the beta 1 receptors on the heart so that it reduces the effect of norepinephrine and epinephrine on your heart. And again, your book has a nice table that talks about agonists and antagonists as well. And there you go. With that, that is everything you are responsible for in Biology 430. All right. Questions on any of that? All right, and with that, we are done. Congratulations. The hard work, well, okay, that's not true. My hard work is done, which is actually not really true either, because I still have to finish writing the exams, because I'll be honest, I haven't finished writing the exams yet. But the important thing is we have now covered all the information you are going to be responsible for on Tuesday's exams. And as someone mentioned in the last class, so again, I'll make sure to emphasize it again. Remember, the final exam is cumulative, so it involves everything, and that includes the nervous system as well. I haven't started even looking at the final yet, but again, in simplest terms, if you think about it, we have had 5 sections to this class. All right. And so likely the final will be something approximately to 5 parts, equal parts of all 5 parts. So again, that sounds about 20 questions, for everything for the directional terms and the cells, and then 20 questions on the integumentary and tissues, 20 questions on muscles, 20 questions on bones, and 20 questions on the nervous system. And again, it might not be exactly that, but it's going to be close, about 20% of the exam on each of our 5 sections, including the nervous system. So you need to know the nervous system both for these coming exams and for the final exam as well. All righty. So any questions on any of that, anything that needs to be covered on the recording or anything like that? I already see some people dropping out. So excellent. I guess that means you guys have got it. All right. So then what I'm going to do, thank you. I appreciate that. Again, I appreciate the kind words, but again, it's been a little crazy. Correct. Great question. So yes, as it's been for the entire class, if there is something that is covered in the book that we do not talk about in the lecture or isn't on one of your other assignments. So for instance, some of the stuff in your Physio X, we may not have explicitly covered in the lecture because I knew you were getting in there. But if there is some piece of material that we haven't covered in any of this, some of the developmental stuff we haven't talked about, other things like that, yes, you are not responsible for. You're responsible for the stuff that either I assigned or the things that we talked about in class. So if there's, and there, yeah, there's always stuff in the book that we don't cover in class, and you're never responsible for that stuff, unless I explicitly say that. And I don't believe I did that for this section. All right. All right, I appreciate that. I appreciate that, guys. Again, it's been a fun class. It's been a weird semester. So this has been a weird semester. So I wish you all the best. Again, I will. So when we do, because I had enough people and I had a couple of things. So what I will do next Thursday is what we'll offer a review. I'll put that on the Confer Zoom schedule so that we can do it. We'll start at the same time. We'll go noon, maybe noon to 2, and then obviously we can stay later if need be. But I will go ahead and put that on there. So if you guys have questions about that, but otherwise between now and Tuesday, if you have questions, always feel free to find me, ask those questions, and then we'll figure it out from there. Good luck, stay safe, and take care. I'll see you guys soon. All right, bye.